Right. Hello, students. Today we are continuing to think about proteins, and our topic today is protein structure and protein function. So, you've seen this diagram before. Lots and lots of different proteins, all with their own different structures. And I hope you remember from last time we saw how each protein has its structure, how the structure is formed, how a chain of amino acids, a polypeptide chain, is able to fold up to make a folded structured protein. And every organism has many different proteins. Each of those proteins has its own function. It has a role to play, a job to do in making that organism work correctly. So, how does a protein go from having a structure, a shape that we know about? How does having that shape allow the protein to carry out its function? Well, that's what we're going to be talking about today. So just to recap what we talked about last time, I hope that you remember, we saw that the primary structure of a polypeptide, of a protein, is a sequence of amino acid residues, which are then able to fold up through different levels of folding: secondary structure, tertiary structure, quaternary structure. But all the information that's needed for forming the final shape, the final form of the protein, comes from the nature of the amino acids in its polypeptide chain. Okay, so that's how a protein gets its structure. How does the structure lead to function? So this is just a few sentences from your curriculum, just to help you to know where we're going today. So in your curriculum, we are supposed to be covering functions of proteins, structure function relationship of proteins, and then next time we'll be looking at functional adaptations of oxygen binding proteins, which is a very big topic with a lot to cover, so I've put that in a separate lecture. So for today, I'm just going to be introducing you to the idea of how the structure of a protein can lead to it correctly being able to carry out its function. And the examples I'm going to be taking today are keratin, which is a fibrous protein, and ovalbumin, which is a globular protein. And then next time we're going to be looking at the extremely important oxygen binding proteins, myoglobin and hemoglobin. Okay, so that's just where we're going over the next two lectures. So starting with keratin. So keratin is the protein that gives hair its shape and texture. So I'm touching my hair now. I think that most of you have probably got hair. If you want to touch your hair, think what it feels like. Think of the shape of each hair, the texture of the hair. That is due to the protein keratin in your hair. Okay, so just some pictures of some hair. I don't know if anyone has hair like this, but I'm sure you all know what hair looks like and feels like. Okay, so keratin is not only found in hair, it's also important in skin. So if you touch your skin, if you feel the way that your skin is a solid layer on the outside of your body, if you perhaps have a sore place where the skin is damaged, you'll know that that feels different to your normal skin. And if we look at this diagram of the skin, you'll see that the outer layer of the skin is called keratinized squamous cells. These are flat cells which are basically just filled with keratin protein, and they form a protective layer on the outside of your skin. And then underneath there are cells which are still actively dividing and functioning. So the outside layer is important for protection, but the cells that form that layer are now more or less just containers for keratin. And it is the keratin that gives the protection on the outside of the skin. I'm sure you'll recognize this sort of animal. We see a lot of them around southwest Uganda. Their horns are again made of keratin. And if you've felt a cow horn, you'll know that it's very hard. You'll know that it's a very rigid substance, it's very solid. Again, that hardness comes from the keratin in the horn. Okay, so that's where we find keratin. So its function is a structural function, a protective function. Its function is to be hard, basically. Okay, so let's go back to thinking about a hair. Here is what a single hair looks like under a microscope. And this is a diagram of a cross-section of a hair. So inside the hair, there are cells, and inside those cells is a lot of keratin. Okay, and this diagram just shows the keratin sort of pulled out, so we can see what it looks like from inside the cell, sort of magnified and pulled out as it comes towards us here. Okay, so what are we seeing? Here we can see an alpha helix. I hope that you're familiar with what I mean by an alpha helix by now. A two-chain coiled coil, a protofilament, a protofibril, an intermediate filament, and then cells. So these are higher levels of association of keratin molecules. So up to this point, we're talking about molecules. Here we're talking about macromolecules, a number of molecules joined together. Okay, so here is a keratin alpha helix. This is an alpha helical secondary structure of keratin protein. Here is a two-chain coiled coil. So that's what we've got here. So in the coiled coil, we have two keratin molecules associating together as subunits of a dimer. Two are coming together and coiling around each other. In a protofilament, a number of these structures associate together side by side. Do you see the way they're lining up side by side to make a higher level structure, a larger structure? And then lots of protofilaments line up side by side to make a protofibril. We're just looking at a flat section here. Remember that we can have more above and below this flat plane as well to make a sort of spherical structure, or a round structure, as we can see here, a tubular structure. Okay, so each keratin molecule has two polypeptide chains, and each of them is an alpha helix. So this is a keratin molecule made from two polypeptide chains, two subunits. These are twisted around each other in a coiled coil. So this is the coiled coil structure It's a coil because of the alpha helix, and it's a coiled coil because it's then coiled again. Okay, so this is what we're seeing. This is what we mean by a coiled coil. Here is one protein alpha helix. Here is a second protein alpha helix. These two helices are wrapped around each other to give a shape like this. So each of the helices is a coil. Wrapped around each other, it becomes a coiled coil. Okay, and remember last time we talked about right and left-handed helices. The alpha helix is a right-handed helix, but if you look at the twisting we see here, you'll see that it's actually a left-handed helix. If you curve your left-hand fingers around and your thumb follows the direction of the coil, you'll see that your left-hand fingers follow the direction the coil is moving, whereas your right-hand fingers don't. Okay, so here's just a little moving diagram so you can see it again. So this emphasizes the coiled nature of each of the alpha helices. You can see the right-handed alpha helix in each of these chains, and then you can see that they're again coiled around each other. We're just showing one turn of the coil here. They're coiled around each other to give the left-handed helix of the coiled coil. Okay, so that's what we're seeing at this stage. This is the coiled coil. These molecules can then associate with each other. They can interact between the sides of the proteins and the ends of a protein. We'll talk about it in more detail in a minute, but notice that the top and the bottom of the protein are slightly different to the middle section. Okay, and these can associate side to side to form filaments, small filaments then building up to big filaments or fibrils. Okay, so this is what each of those molecules looks like. Here is the dimer made up of two keratin molecules twisted around each other. Okay, so the middle part here is all alpha helix. So you can see in each molecule, there's a green molecule and a purple molecule shown here. So the green molecule twists around and its alpha is alpha helix. The purple molecule twists around and is alpha helix. And then these two helices are twisted around each other in a coiled coil. Okay. So if we want to talk about this molecule in terms of the levels of protein structure we talked about last time, the alpha helix is, of course, secondary structure, but the whole molecule also has a head region and a tail region. Okay, so this represents a form of tertiary structure. So even though it's such a long molecule with just a folded head at one end and a tail at the other, this is its tertiary structure, made up mostly of secondary structure alpha helix. Then when the two molecules come together to make the coiled coil dimer, that is an example of quaternary structure, two subunits that make up the final protein in its active form is a form of secondary structure. A form of quaternary structure. Okay. So how do the different molecules make the filaments? So we can have associations at the ends and at the sides. So if you look at this diagram in the protofilament, the molecules line up in a sort of staggered fashion. They've not got their heads together and they've not got their tails together. And here we've got one line lined up in this way. We've got interactions between the head of one and the tail of the next. And the next filament is going in the opposite direction with interactions between its head and its tail. And you can see in this diagram how these structures fit in to a protofibril. So we've got interactions. These are non-covalent, weak interactions, if you like. But again, as we keep saying, many weak interactions can actually form in total a strong interaction. So interactions top to bottom and interactions side to side to line up the dimers, the keratin dimers, to make a filament. Then lots of fibrils line up together to make a microfibril. Okay, layer after layer after layer of structure getting bigger and bigger at each stage until finally we have basically one of the hairs on our head, which has lots of different cells inside, which are packed with these different fibrils of keratin. Okay, so yesterday or last time I talked about protein databases and the fact that we can use protein databases to find protein sequence of any protein that has been entered into it, hundreds and thousands of them. So I went to a protein database and found the primary structure of a human keratin. I say a human keratin because we have a number of different genes coding for slightly different keratin proteins. We have different keratin in our hair and in our skin and in other parts of our body. They're all keratin, but there are slight differences. So this is one of the human keratins. And I used the prediction software in protein database to predict whether or not a particular amino acid sequence is likely to form secondary structure. And this is what we find for keratin. So in gray here is a region which is not likely to form secondary structure. Underlined in red is a region that is likely to form alpha helix. Okay, so you can see that most of the molecule is very likely to form alpha helix, predicted by computer modeling. And this is exactly what we know from our examination of the actual structure of keratin. Most of it is alpha helix with a different bit at the top and at the bottom. In this particular form of keratin, the bit that isn't helix at the end is only 3 amino acids. Okay, so here, the primary structure, the sequence of amino acids, is what is important for making the correct secondary structure, which is alpha helix. And that is exactly the way this protein should be. So the primary structure determines the secondary structure. So how do the two molecules of the dimer come together? Well, within that sequence of amino acids, there are a number of hydrophobic amino acids. So I hope you remember the hydrophobic amino acids, the ones which don't like being in contact with water. They like to hide away, particularly alanine, valine, leucine, isoleucine, methionine, and phenylalanine. Okay, so if we look at this diagram, we can see that there are quite a lot of leucines, there are quite a lot of isoleucines, there are quite a lot of phenylalanines, and so on. And when the protein, when the helix, when the polypeptide chain is coiled up into an alpha helix, these hydrophobic amino acids end up making a sort of stripe along one edge of the alpha helix. And it's not a straight stripe, it's a slightly, if you don't become confused by all the helices, it's a stripe that forms a helix around the helix. Okay, can you see that? This red portion in the diagram represents the hydrophobic amino acids and where they lie in the alpha helix of keratin. So when two chains of alpha helix in two different keratin molecules come together, those hydrophobic amino acids want to hide away from water. So the most energetically stable form for these two chains to interact together is for the hydrophobic amino acid stripe to sort of stick to the one on the other alpha helix. Okay, so you can see the association is starting here at one end, and then because of the way the stripe curves around the helix, that brings the two chains of alpha helix in the protein together to coil around each other in this way. Okay, can you see that? So hydrophobic interactions trying to get away from water, making the most stable form of the protein to be one where water is excluded from being able to get into contact with the hydrophobic residues, means that one of these chains of alpha helix wraps around the other in the coiled coil. And this just shows a wireframe diagram, again, the hydrophobic residues being highlighted in red here to show how they can form interactions to get away from the water molecules. So in this way, the primary structure, so the positioning of each of these hydrophobic residues, is determined by the primary structure, okay, the amino acid sequence in the protein. The primary structure determines, the primary structure makes the quaternary structure. The interactions between the two subunits happen because of the positioning of hydrophobic amino acids in the primary sequence. Okay, so primary structure determines secondary structure, primary structure determines quaternary structure. So how does this relate to function? Well, the other important thing we see about keratin is that it can also form sulfur bridges between the two different protofilaments. So here are different protofilaments, and here represented in yellow are sulfur bridges, disulfide bonds. And the more disulfide bonds a protein is able to form, the harder the protein is. So I said that there are a number of different sorts of keratin. One of the big differences we see in keratins with different functions found in different parts of the body is the number of cysteine residues and therefore the number of cysteine disulfide bridges that can be formed. So hard keratin, for instance, in those cattle horns we saw, has many sulfur bridges. Okay, so again, the positioning and the presence of cysteine residues able to form sulfur bridges is part of the primary structure of the protein. But the primary structure, the particular amino acids that are present, allows the protein to carry out its correct function. Okay, so the primary structure of the keratin relates to the function of the protein, making it hard. So next time you're out walking along the road, perhaps, and you see one of these animals with these magnificent horns, I want you to look at it and think, wow, what a lot of sulfur bridges. Okay, will you remember that? When you think of hard keratin, think of sulfur bridges and think of cattle horn. Okay, so let's think a bit more about these sulfur bridges. We saw last time that sulfur bridges can be broken by being reduced and can be formed by being oxidized. And this can be made use of in the process of hair relaxing or permanent waving. These two are more or less the same thing, but hair relaxing aims to make curly hair straight, or permanent waving aims to make straight hair curly. I don't know why people are never satisfied with the hair that God has given them, but people with curly hair seem to want straight hair, and people with straight hair seem to want curly hair. And this can be done by a chemical process. It's biochemical engineering on hair. So by adding a reducing agent to the hair, the sulfur bonds, the disulfide bonds, sulfur bridges can be broken, hydrogen's added back to them, they can be reduced, and that allows the keratin microfibrils to slide along each other and change shape. Okay, and then when they've changed their shape, the sulfur bridges can be oxidized again to hold them into the new shape. So I think that most people in Uganda are more interested in making curled hair straight, so you'd be going in this direction. So the sulfur bridges in the curled hair would be reduced, broken apart like this. The hair would be physically straightened out, and then an oxidation reaction would reform the sulfur bridges, and the hair would be held in the straightened conformation, in the straight shape. Okay, so permanent waving is going in this direction, so reducing the bridges in a straight hair, physically curling the hair around and oxidizing so that the sulfur bridges reform and hold the hair in a curled shape. Okay, so that's hair relaxing, and I don't know if you've ever smelt what it smells like when the chemicals are working on hair, you can often smell a very strong smell of sulfur, a smell of bad eggs, if you like, a smell that's coming from these disulfide bonds being broken, and hydrogen sulfide can also be released by this process and gives that strong smell. Okay, so hair can be relaxed or waved by breaking and remaking disulfide bonds, sulfur bridges. Okay, so just to summarize what I've said about keratin, the primary structure has a high tendency to form alpha helix, regularly spaced and twisted hydrophobic residues forming a stripe along that alpha helix causes the formation of the coiled coil quaternary structure, and many cysteine residues allow the formation of sulfur bridges to cross-link the filaments. So keratin, because of its long, thin nature, is classified as a fibrous protein, and it's an important structural protein in human beings and in cattle and in many, many other organisms. Okay, a pause for questions. Okay, so I said that keratin is an example of a fibrous protein. There are many other sorts of fibrous protein as well, and most of them are structural proteins. That means they're proteins that are important in allowing our cells and therefore our whole bodies as a multicellular organism. They're important in giving our cells and our organs and our bodies their shape and their structure. Another important example of a fibrous protein is collagen, which is important in tendons and cartilage and bone. And here's just a diagram of collagen. It's a right-handed coiled coil of 3 polypeptide chains. Okay, so here is a wireframe model emphasizing an alpha helical shape, and here is a space filling model of a single chain and the trimer, the 3 chains wound around each other. And I want you to find out about collagen for yourself. Last time I suggested that you might go to the protein data bank and read about it. You can also read about it in Leninger textbook. So please look up collagen, research collagen, learn about collagen for yourself. Okay. So we tend to classify proteins in 2 general classes as fibrous or globular based on their tertiary structure. So if we think about fibrous proteins, keratin is one example, collagen is another. Polypeptide chains are arranged in long strands or sheets. The tertiary structure tends to be quite simple. We saw for keratin, but it's a head and a tail with an alpha helix in the middle. The monomers are usually similar. They are not soluble. Now, this is important. If you think about your skin or your hair, it doesn't dissolve, does it? It would be very difficult to wash ourselves if it did. So it's good that our proteins will not dissociate and dissolve in water. Okay, our skin remains covering our body even if it gets wet. So this allows for the function of fibrous proteins to be structural, providing shape and support and protection to our cells and to our organs and our bodies. Okay, so the other classification of proteins as globular proteins is slightly different. So here, the polypeptide chains are folded into a spherical shape or a globular shape. They're round rather than long and thin. The tertiary structure tends to be complex, and the monomers are often, not always, but often different. And they are soluble. They are able to be dissolved in water. So these are the sort of proteins that we find dissolved in the cytoplasm of our cells, inside our cells. That association with water is necessary for them to have the right shape and for them to be able to carry out their correct function. And their functions are, well, many, many different functions of proteins. They can be enzymes, they can be regulatory proteins, they can be storage proteins, many, many different functions. Okay, so the example I'm going to give you for for a globular protein is ovalbumin. So I think this is a protein that you're already very familiar with, although you might not know it by that name. What's that? I hope you're all saying to yourself, it's an egg, obviously. It's a hen's egg. And you know that if you break that egg open, you see something like this. So this part of the egg, forget about the yolk for a minute, but this part of the egg is the white of the egg, although in this form it's not white, it's transparent. This is made up of about 90% water and 10% protein dissolved in that water. And most of that dissolved protein is ovalbumin. Okay, so that's where we find ovalbumin. And I think that you already know what this is. Okay, what is the difference between an egg that looks like this and an egg that looks like this? I hope that you know that the difference is applying heat. You take an egg, you break it into a pan, and it looks like this. You apply heat, and it starts to look like this. Or you can take an egg like this, put it in boiling water for a while, take the shell off, and it looks like this. Okay, so we've changed from being a transparent dissolved solution of protein to a white solid. Okay, so what is happening when you apply heat to the white of an egg? Well, I've again gone to a protein data bank and found the primary structure of ovalbumin, and here it is. It has 386 amino acids, just written out here with one letter abbreviation code divided up into groups of 10 to make it easier to count and to follow, and then on lines labeled with the number. So from 1, which is a methionine, to number 386, which is a proline. Okay, so 386 amino acids, and if again we predict the secondary structure, you'll see that it's a lot more complex than the prediction that we saw for keratin. Here we see different sorts of secondary structure. Helix, shown in blue, So this just represents the whole of these 386 amino acids from 1 to 386. So helix is shown in blue. Beta strand, that's one strand that's likely to form into a beta sheet, is shown in green. And then another form of secondary structure that I didn't really talk about, a turn or a beta turn, is shown in pink. That's a place where the polypeptide chain is very likely to form a very sharp turn to go in a different direction. Okay, so that's the prediction of secondary structure from the primary structure, the amino acid sequence. And this is a diagram showing that prediction of secondary structure lined up along the amino acid sequence of an ovalbumin molecule. Okay, so you can see here again alpha helix shown as a red coil and beta sheet shown as a yellow arrow. And here are the turns shown in purple. Okay, so this is the secondary structure prediction for ovalbumin. Those elements of secondary structure are able to fold up into a tertiary structure, which looks like this. So you can see the alpha helices here have sort of come together to associate and interact with portions of beta sheet. Okay, so the protein is now folded up into its tertiary structure because of the interactions between the parts of secondary structure. Okay, and then the whole ovalbumin molecule is actually a tetramer. It has 4 different subunits. So the quaternary structure is a whole protein molecule of ovalbumin which has 4 identical subunits. Okay, remember that subunits can be the same or can be different. In this case, they're all the same. They're all identical. They all look like this one. So here is the whole molecule. This just shows how one subunit it can be part of it. So we've got one subunit shown in pink, yellow, blue, and green. And the 4 come together and interact together to make the whole ovalbumin molecule. Okay, so there are the levels of primary protein structure, again, primary, secondary, tertiary, and quaternary structure. Okay, so let's think about some of the properties of ovalbumin. So first of all, it's soluble. Remember that we said that the egg white is 90% water, but I think if you think of feeling an egg white between your fingers, you know that it doesn't feel like water. It feels slippery and a little bit sticky, yes? So egg white is 90% water, but the 10% of protein that's dissolved in that water changes the nature of the solution. So it's a solution of protein, mostly ovalbumin, in water. And it's quite soluble. So 10% of the total mass of egg white is protein. So the other thing we know about ovalbumin is that it's denatured by heat. Its normal nature, its normal form that we find when we break open an egg, is changed when it's heated. The protein becomes denatured and changes its form to a different form. So what's happening actually on a molecular level when the protein becomes denatured? Well, the quaternary, tertiary, and secondary structure becomes lost because the heat disrupts the non-covalent bonds that are holding the molecule together. Okay, so if I go back to this diagram, you'll remember that all these elements of secondary structure are held together by non-covalent bonds, by hydrogen bonds, by van der Waals forces, by ionic bonds, and so on. So the structure is held together by non-covalent bonds. When it's heated up, the peptide bonds that form the amino acid chain are not damaged by the amount of heat that it needs to cook an egg. But all the covalent bonds that help it to form this structure they're broken, they're denatured by heat. So those polypeptide chains are then become loose, they move around in relation to each other. And every now and again, two cysteine molecules that don't usually associate together will happen to bump into each other and dimerize, form a sulfur bridge, and form a cysteine molecule. So that causes a hardening of the protein. Okay, you'll know that egg white is a lot harder than cooked egg white is a lot harder than raw egg white. So denaturing the proteins, making them lose their normal native non-covalent bonding, allows them to form non-specific bonds, especially sulfur bridges, which causes the protein to become hard. Okay, so I'm going to show you a video now. This is quite a fun video. You can find it on YouTube, and I hope you'll find it interesting.
It's so obvious that it's practically proverbial. You can't unboil an egg. Well, it turns out you can. Sort of. What thermal energy does to the egg's molecules, mechanical energy can undo. Eggs are mostly made of water and proteins. The proteins start off folded up into intricate shapes, held together by weak chemical bonds. Adding heat disrupts those bonds, allowing the proteins to unfold, uncoil, unwind, and wiggle freely. This process is called denaturing. The newly liberated proteins bump up against their neighbors and start to form new bonds with each other, more and more as the heat increases, until finally they're so entangled that they gel into a solid mass, a boiled egg. That entanglement might look permanent, but it's not. According to a chemical idea called the principle of microscopic reversibility, anything that happens, like egg proteins seizing up, can theoretically unhappen if you retrace your steps. But adding more heat will tangle the proteins further, and cooling them down will only freeze them. So here's the trick. Spin them around ridiculously fast. I'm not kidding. Here's how it works. First, scientists dissolve boiled egg whites in water with a chemical called urea, a small molecule that acts as a lubricant, coating the protein's long strands and making it easier for them to glide past each other. Then, they spin that solution in a glass tube at a breakneck 5,000 rotations per minute, making the solution spread out into a thin film. Here's the key part. The solution nearest the wall spins faster than the solution closer to the middle. That difference in velocity creates shear stresses that repeatedly stretch and contract the proteins until eventually they snap back into their native shapes and stay there. By the time the centrifuge stops spinning, the egg white is back in its original unboiled state. This technique works with all sorts of proteins. Bigger, messier proteins can be more resistant to being pulled apart, So scientists attach a plastic bead to one end that adds extra stress and encourages it to fold up first. This unboiling method won't work with a whole egg in its shell, since the solution has to spread throughout a cylindrical chamber. But the applications go way beyond uncooking your breakfast, anyhow. Many pharmaceuticals consist of proteins that are extremely expensive to produce, partly because they get stuck in tangled up aggregates, just like cooked egg whites, and have to be untangled and refolded before they can do their jobs. This spinning technique has the potential to be an easier, cheaper, and quicker method than other ways to refold proteins, so it may allow new drugs to be made available to more people faster. And there's one more thing you need to keep in mind before trying to uncook all of your food. Boiling an egg is actually an unusual cooking process, because even though it changes the way proteins are shaped and bound together, it doesn't actually change their chemical identity. Most types of cooking are more like the famous Maillard reaction, which makes chemical changes that turn sugars and proteins into delicious caramel crunchiness, and are a lot harder to undo. So you might be able to unboil your egg, but I'm sorry to say you can't unfry it yet.
Okay, I hope you enjoyed that video. I thought it was quite useful for illustrating some of the things we've been talking about. And also, it's a technique, it's a real technique that was only developed about 4 or 5 years ago now. So it's a new technique that I think might be becoming more and more used in pharmaceutical processing and so on. So it's something that you might be coming across in the future. Okay, so denaturing protein and possibly renaturing it again doesn't only happen because of heat. It can also happen by changing other things about the physical conditions that the protein is in. And I've got an example here of using pH to denature ovalbumin. Okay, so a little video again. So cracking eggs into a beaker, collecting the egg white, we don't want the yolk, and then adding strong acid, strong hydrochloric acid. And you can see that it's changed color, and stirring it, encouraging that tangling. And now something solid, looking rather like cooked egg white, but in this case, we don't have any heat. It's changing the pH, and changing the pH around the protein has a similar effect of breaking the non-covalent bonds, making the polypeptide chains open up, move around, especially with the agitation of the stirring motion, and become associated with each other, forming non-specific bonds and therefore becoming a hard mass of protein. Should we watch again? Okay. Collecting the ovalbumin solution. Adding acid. Agitating the solution to encourage the protein to unfold. And demonstrating that it's become a hard mass, showing non-specific covalent interactions between the polypeptide chains. Okay. So let's have a think now about how this structure of albumin allows it to carry out its function. Well, first of all, what is the function of albumin? Why do we find albumin dissolved inside eggs? Well, one of its functions is to form a protective gel around the developing chick embryo. Okay, it's a thick solution. If the egg is moved around, if a hen is incubating her eggs and turns them around, having this thick liquid around the developing embryo means that the embryo isn't damaged by being moved around in that way, by being shaken or even rolled around a bit. Okay. Right. It also provides a source of nutrients for the growing chick embryo. So here is a diagram of a 5-day-old chick embryo developing inside the egg. Here is the yolk, here is the white of the egg, and here is the developing embryo. And if you've ever seen a fully grown chick hatch out of its egg, you'll know that no liquid is left inside the egg. It's just chick by the time it hatches. And that's because all the yolk and the white provide all the nutrients that are needed to become the body of the chick. And that includes the ovalbumin protein dissolved in the egg white. So it provides a source of nutrients for the growing embryo. It's also a calcium binding protein. I didn't point it out, but if I go back to the diagram here, you can see little space filling molecules on our ribbon diagram here. And each of these is responsible for binding calcium ions, and a high calcium concentration able to reach the growing embryo would be damaging to the growing embryo, would cause it to not form correctly. But the eggshell, the shell of the egg, is made from calcium carbonate, so which could dissolve in the albumin and reach the growing chick. So the calcium binding properties of ovalbumin protein protect the chick from excess concentrations of calcium disrupting its growth. Okay, so that's the function of ovalbumin. Okay, so a pause for any questions. And just a recap of what we've talked about. They are the definitions I gave you last time of the different levels of protein structure. So if you're a person who likes learning definitions, please do learn those definitions. They may be useful for you. And I hope that they help you to understand how protein structure can allow a protein to carry out its function. Next time, we're going to continue with this idea as we look at myoglobin and hemoglobin. Okay, so I just wanted to say a few words about some other words that are used to describe protein structure that you might come across as you look in the textbook. I'm going to talk about protein domains and protein motifs. Okay, so a protein domain is a name given to one part of a protein that tends to have a particular structure and function. Okay, so here is a diagram, a space-filling diagram of a globular protein, and here is the ribbon diagram of the same protein. And labeled here, there are two domains, an SH2 domain and an SH3 domain. So each of these domains, you can see it's sort of a fold here joined onto a fold here just by a polypeptide chain. We're not actually associating with each other, just joined by the chain inside them. And this is a common feature of a domain. It tends to have its own tertiary structure, and it can often fold into the correct structure even if it's separated from the rest of the protein. So if we were to cut this polypeptide chain at this point and isolate this part of it, it would still have the ability to fold into this structure. It isn't relying on all the rest of the protein to be able to fold up. Okay, and the same with the SH2 domain here. So protein domains can form their own tertiary structure. Sometimes a structure which is a domain of one protein also has its own function as a separate protein. So here is the protein pyruvate kinase, an important enzyme. And you'll see in the center of this protein, there is a structure that's labeled a TIM barrel, TIM barrel, made up of a number of alpha helices lying together in a sort of barrel shape, a circular shape, And each of the parts of that barrel is made up of a domain which is the same as triose phosphate isomerase, or TIM. Okay, so you see that this structure here is the same as this structure here. Here it's a protein in its own right. It's another enzyme, an isomerase enzyme by itself. Here it's a domain of a larger protein, pyruvate kinase. Okay, so a TIM can be a protein by itself, or it can be part of a larger protein. And this means that we can make proteins up by putting domains together, or I should say proteins are made up of different domains. So here is an example of a number of proteins that are made of different domains joined together. So here is a protein called EGF, which has a domain which is represented by a green hexagon here. Here's chymotrypsin, a digestive enzyme that you might have heard of, but has a domain represented by a brown oval shape. Okay, if we look at the protein urokinase, we'll see that it has one domain that's the same as EGF, another domain that's the same as chymotrypsin, but then a third domain in between them, which is different. But if we look at plasminogen, we can see that plasminogen has a number of the same domain. And here's factor IX, factor 9, which has the brown domain, the green domain, and another different one, a yellow triangle. Okay, so these different domains with their own different structures and different functions can be joined up together to make new proteins with different properties. Okay, so if you hear about the word domain being used, we'll be talking about something that carries out a particular function. For instance, next semester, you will probably be hearing about proteins with DNA binding domains. That means part of a protein is good at binding to DNA, but the rest of the protein, once it's bound to the DNA, might be doing a number of different things, but one domain allows the protein to bind to DNA. Okay, so that's domains. And we've talked about databases. A very common investigation that can be done using protein databases is to look for domains within proteins. So here you'll see an alignment, a number of proteins, primary sequence, amino acid sequences lined up together, being searched for what's called signature sequences. They are sequences which identify a particular type of domain. So here is a protein from a human. Here is a protein from a fruit fly Drosophila, and lots of differences in the protein, but parts of them can be used to identify similar domains by matching the amino acid sequence. Okay, so this is what's known as homologous domains, similar domains in different proteins. Okay, we'll be thinking more about that sort of idea in molecular biology next semester, but I'm just mentioning it now because you might come across the word domain if you're reading textbooks or internet. Okay, and another similar word that you might come across is a motif. This is something that's similar to a domain, but it's not usually a separate part of a protein in the same way that a domain is. It's a pattern, a structure that's found in many different proteins which allow them to carry out a similar function. So the example I've taken here is again DNA binding, but this is a DNA binding motif, which is called a helix turn helix motif. Okay, and it's a protein that has just the right shape to bind to DNA by fitting into the side of the DNA molecule. We'll talk about that more in a few weeks' time when we look at nucleic acids. So motifs are often talked about by their function. So we might have a DNA binding motif as we have here, and previously we saw a calcium binding motif. Okay, so motifs can be found in different proteins, but carrying out a similar function. Okay, so you can think about any questions you might have on that. And I'm really coming to an end for today. Just to remind you about textbooks again, if you're going to hard copy in the library, you'll want to look at chapter 6, which is about proteins, or if you've got the 2008 edition, the same material split into the 3 chapters, chapters 3, 4, and 5. Okay, and some of you might have a more modern soft copy. Again, it's chapters 3, 4, and 5, cover proteins. So that's all for today, and next time we're going to go on and think about oxygen binding proteins.